Okay, let's dive right into this. Welcome to this explainer, where we're going to tackle what is arguably one of the most profound biological mysteries out there, human consciousness. Think about it. How exactly does a kilogram of disconnected, squishy cells produce the seamless, unified stream of reality that you are experiencing right at this very second? It's kind of wild when you pause and really consider it. Today, we're going to explore a fascinating hypothesis from the world of neuroscience, drawn directly from our source material, that suggests the answer might actually just be a rhythm. To really understand the scale of this mystery, I want you to actively do something for me. Right now, just hold an image in your mind. It could be a scene, maybe a memory from this morning, or even just look around the room you're currently in. It feels like a continuous whole movie playing seamlessly, right? It honestly feels like there is a single observer, just sitting in a little theater inside your skull, watching this beautifully finished picture on some sort of inner screen. But here's the paradox that plunges us straight into the mystery of our own brains. That inner screen, it doesn't actually exist.
Let's break this down with a classic example. Picture a red rolling ball. When you look at that ball, your brain doesn't process it in one single spot. Instead, your vision is literally carved up. The color of the ball is processed in one specialized patch of your brain. Its shape, that's processed in another entirely different area. And its motion across the floor is handled by a third. All of this heavy lifting is done by different neurons that literally never touch each other. And the deeper neuroscientists look into the brain, the more fragmented it appears. There's just no final location where all these reports come back together. And yet, you never see a disembodied redness just floating beside a shapeless motion. You effortlessly experience one unified object. The features are bound together perfectly. In philosophy and neuroscience, this is what's known as the binding problem. How does the brain stitch these scattered perceptual pieces into a single, seamless moment of unified experience? Any theory of consciousness absolutely must solve this fundamental illusion first, because we take it for granted every waking second of our lives. If the parts are processed separately, what exactly is the glue?
Section 1. The 40 Hz solution, binding by synchrony. So the crucial point is this. To solve this massive biological problem, neuroscience has proposed an incredibly elegant solution over the last few decades, and it revolves around this number, 40, roughly 40 beats a second. The proposed glue holding your reality together isn't a physical place in the brain, it's a rhythm. It's time, not space. You see, when neurons in completely different parts of the brain process features of the exact same object, they synchronize their firing. They oscillate together in what's called the gamma band, at roughly 40 Hz. By firing together at exactly the same time, the brain basically tags these scattered pieces with a temporal signature. It's like the brain's way of saying, hey, these things go together. And the scientific detective story behind this is fascinating. It starts back in 1989, when Charles Gray and Wolf Singer first discovered this synchrony in the visual cortex of cats. That was a massive clue. Then, in 1990, Francis Crick, the co-discoverer of DNA, and Christoph Koch made a really bold claim. They linked this 40 Hz synchrony directly to conscious awareness itself. But I really want to highlight the 2007 breakthrough by Lucia Melloni. Her team ran this completely brilliant experiment. They showed people words at the very edge of visibility. Sometimes the people consciously perceived them, sometimes they didn't. What they found was that both seen and unseen words triggered a local brain response. But, and this is the kicker, only the consciously perceived words triggered a long-range synchronization of gamma across widely separated brain regions. That long-range synchrony was the concrete neural signature of conscious awareness. Section 2. The brain's pacemaker, parvalbumin cells. Let's move to and see how this builds. Because if gamma waves are the rhythm of consciousness, we absolutely have to ask what exactly is generating this rhythm. We're shifting our focus now from the theory to the actual biological machinery inside your head. For a long time, researchers only knew that gamma accompanied perception. They couldn't prove it caused anything. But that changed in 2009 thanks to breakthroughs in optogenetics, a technique which essentially uses light to control specific neurons. This led to the discovery of parvalbumin, or PV, cells. Interestingly enough, the brain doesn't generate this fast gamma rhythm with its main excitatory workhorse cells. Instead, it uses these highly specific fast-spiking inhibitory cells. These PV cells act basically like a biological metronome. They blanket the surrounding brain network with incredibly fast rhythmic silencing pulses. By driving these specific cells with light, researchers proved they could generate gamma waves on demand. They literally found the brain's pacemaker. To put that 40 Hz into perspective, a single gamma cycle lasts about 25 milliseconds.
Now, a 25 millisecond window is incredibly brief, but it's just long enough for signals from distant regions of the brain to arrive and be compared, yet short enough to keep your perception fluid and continuous. Neuroscientist Pascal Fries argues that this is exactly how the brain dynamically routes information. If two brain regions oscillate in phase, they can talk to each other efficiently, because one sends a signal exactly when the other's 25 millisecond window is open to receive it. It's effectively a just-in-time communication network that is opening and closing dynamically.
Section 3. Altering the rhythm. Psychedelics and gamma. So, if this 40 Hz synchrony is the machinery of conscious integration, then introducing specific substances should alter our experience, right? Let's look at what happens when this delicate rhythm is chemically perturbed. And this brilliantly illustrates that consciousness isn't just a simple volume dial you can turn up or down. Let's compare two different mechanisms. On one side, ketamine blocks NMDA receptors on those pacemaking PV cells we just talked about. This disinhibits the cortex and paradoxically creates a massive surge in gamma power. But it's a chaotic, disordered state. The gamma is elevated, sure, but it's coupled to the wrong things, which leads to dissociation. On the other hand, classic psychedelics like psilocybin cause a broadband desynchronization. The tight, organized rhythmic scaffolding of the brain essentially drops away, and the activity wanders freely. In both cases, the unity of ordinary experience softens or fragments. The key takeaway here? Consciousness requires gamma in harmonious, ordered relationships. It's all about binding the right things together at the exact right time, not just having louder gamma waves.
Now, any good explainer must include the scientific caveats, and the source material is very clear about these honest cautions, so we need to be too. First off, correlation is not identity. Just because gamma tracks conscious perception doesn't automatically mean it is consciousness. The binding by synchrony hypothesis is highly influential, no doubt, but rival theories definitely exist. There are also methodological debates, like whether high-frequency EEG readings might sometimes just be tiny eye muscle artifacts picking up on the scan. Clinically speaking, we know the system is fragile. Parvalbumin cells and gamma rhythms are notably disrupted in conditions like schizophrenia, and perturbing this delicate system with the substances we just mentioned carries real serious risks for people who might be vulnerable to psychosis. So keep in mind, this is an active, heavily debated frontier of research, not entirely settled fact. Section 4. The music of consciousness, a fleeting agreement. Now, what's really interesting here is how we conceptualize all of this complex biology. To bring this all into one cohesive picture, I want to read a quote directly from the source material, because it is just a stunning way to frame everything we've covered today. It says, "Consciousness may be less like a picture painted in some central studio and more like music. Not any single note, but the synchrony among them, the fleeting agreement of distant players to sound as one. Your brain doesn't have a central conductor. There's no one up at a podium. Instead, millions of distant, highly specialized cells must constantly agree, millisecond by millisecond, to beat at exactly the same 40 Hz tempo. When they do, the music plays, and the world arrives to you seamless, whole, and completely unified." So what does this all mean for us? Well, the next time you look around the room or hold an image in your mind, like we did at the very start of this explainer, I want you to ask yourself this. Is your seamless ironclad reality actually just a fragile 40 Hz symphony of distant cells, all temporarily agreeing to beat as one? It kind of changes how you view every single moment of your life, doesn't it? Thank you so much for exploring the intricate mechanics of the mind with us today. Keep asking questions and keep marveling at that absolutely incredible biological illusion happening right now inside your own head.